Good morning, everyone. Thank you very much for coming to Invited Session 1, whose title is New Frontiers for Experimental Design. I'm delighted to see so many people. I don't think I've ever been in an auditorium as large as this for an experimental design talk. At this rate, we may even break into triple figures, which would be terrific. We have three speakers today: Cathy Ruggiero from the University of Auckland in New Zealand, David Woods from the University of Southampton in the United Kingdom, and Alison Smith from the University of Wollongong in Australia. And then I'll make a few remarks at the end. I should alert you to the fact that this session is actually being recorded. The IBS has decided that it's going to do an experimental trial of recording, I understand, 2 sessions, which will then ultimately go up on the IBS webpage for viewing by a much, much wider audience. So just please be aware of that. If for any reason you don't want to appear on the video, maybe you should hold your question till after the session is over. Anyway, that's enough from me for the moment, so I'll now ask our first speaker, Cathy Ruggiero, to come up.
Can you see me? Not very tall. Although I feel fairly tall here in Italy, whereas in New Zealand I'm in the lower tail of the women's height distribution. Okay, is that okay? Okay, so in my title I use the word full replication. I say when full replication of factorial treatments is not possible, what I really mean is that an integer multiple of full replication is impossible, and I'll explain what I mean by that shortly. So I'm going to actually motivate what I'm talking about today with a real clinical situation. I'm then going to, this particular clinical situation, in terms of experimental design, requires 2 phases of experimentation, and I'll explain what I mean by that. I'll then discuss the particular study that I'm collaborating on with clinicians back in Auckland, and I'll describe the phase 1 and phase 2 experiments in a bit of detail. And I'll look at, or talk about some of the design considerations for this particular setting. Even though it's a small experiment, it's quite large in terms of presenting it here on a screen. So I'm going to look at a slightly smaller experimental design, but it's essentially exactly the same setting. I'll look at the small phase, the phase 1 experiment in that smaller setting, and then discuss some of the ideas about what makes a good design amongst competing designs of the same size. And I'll do this by looking at the theoretical analysis of variance tables, looking at expected mean squares and average efficiency factors of the main effects and interactions. And then I'll look at how we take that material and we assign it to the block structures or blocks in the second phase experiment, and I'll then make some final remarks about what I've talked about. So, oops, that's not where it's meant to, sorry. Okay. So the question my collaborators were interested in was, or is, how does obesity alter the molecular and biochemical responses to a severe acute illness over time? And a very interesting illness to them is sepsis. Now, what happens with sepsis is that we start off with a localized infection here in the lung. Those toxins from that infection then spread and go into the circulatory system. Those toxins then cause, in the circulatory system, then cause a global inflammatory response. That inflammatory response then affects other organs, causing them to function poor or deteriorate and eventually fail. And in a small proportion of people who get sepsis, they ultimately die and nothing can be done about it. So it's quite interesting. It's something that's quite important to understand. The organ that we're interested in looking at is the liver, And the reason for this is it's known to actually, its transcriptome and its proteome are known to change at a molecular level. And for those of you who aren't familiar with transcriptomics, proteomics and metabolomics experiments, essentially we start off with our genetic blueprint, the genome. Some of those genes are actually protein encoding genes. They're referred to as the transcriptome. Those transcripts or messenger RNAs are able to traverse through the cell's wall, the nucleus of the cell's wall, and then they're transcribed into proteins. These are the workhorses of the cell, and of course there are other enzymatic reactions. These are the metabolites. These are smaller molecules than proteins, and they're meant to be more closely related to the phenotype. So in our case, the phenotype will be the disease etiology. So the liver is also metabolically very active. So we're interested in looking at the transcriptome, the proteome, and the metabolome simultaneously. And remember that we're interested in how the disease progresses through time, so we need an organ that can be repeatedly sampled through time and not kill our organism. So the particular organism, I'll tell you about, the organism we're looking at will be the rat. So why two-phase experiments? Well, two-phase experiments arise when the thing we can't measure directly, the thing we want to measure can't be measured directly from our experimental unit. So the phase 1 experiment involves the application of treatments to our subjects or our experimental units, and in this case we're going to take liver biopsies. We're going to take a piece of liver from the same individual repeatedly through time. We can't measure the effects at a molecular level at phase 1, so we need to conduct a subsequent experiment, a laboratory-based experiment, to acquire the data. And this process, and I'll tell you a little bit more about this process, but essentially what happens is, and I'm going to focus on proteomics only, that's all I have time for, involves isobarically labelling peptide samples, pooling them and subjecting them to liquid chromatography and tandem mass spectrometry analysis. Now, a lot of work's been done. The initial work on two-phase experiments was done by McIntyre back in 1955, but a lot of developmental work has been done by Bryan and Bailey in recent years. So, let's consider our study. What we have is a 2x2 factorial with repeated measurements. The two factors are surgery, so a sham surgery where the animal is anaesthetised and then opened and left like that. The sepsis surgery, they actually perform a certain surgery to make the animal ill after the animal's been opened. Now, these animals were previously either on a chow diet, which is the usual diet, so they stay their normal size, or they're on a diet that causes obesity. Okay, so we have lean and obese and sham and sepsis surgeries. That's our 2x2 factorial. And each of those animals has 5 lobes taken from them, or 6 lobe biopsies, one at baseline, before the sepsis or before the surgery, sham or sepsis surgery is performed, and then hourly thereafter, for 5 hours, a lobe biopsy is taken. So for those of you who don't like to look at these things, you might want to close. I should have said that before I showed you this slide. Apologies. But the rat's liver is quite different to the human liver. It actually has multiple lobes. So we see here that there's the superior caudate lobe. It's like these little bits. And so I can't move this mouse very easily, my apologies, but you can basically see the suturing, and they basically tie off a little bit of lobe from each lobe hourly. Okay, so we're looking at rats because clearly it wouldn't be ethical to do this in humans. So we now have our tissue, but we have to take this tissue and we have to now measure the proteins, identify the proteins in the samples and measure the abundances of those proteins. There's a technology called ITRAC, isobaric tags for absolute and relative and absolute quantitation. Essentially what happens is that the proteins are broken up into smaller components, peptides, and then there are these isobaric labels that attach themselves to certain amino acids in the peptides, lysine and arginine. and also at the start of the peptide. Now, because this is the four-plex system, the initial one that was introduced back in 2004. Now, if you have a look at the sum of these tags, the weights, this is the iron and this is the balance group, they all add up to 145 Daltons, okay? So when they go through the mass spec for analysis, these, the same peptide, differentially labelled, say, with 115, will be treated in a homogeneous fashion. Okay, so here is the same set of peptides from 4 different samples or treatment groups, say, differentially labelled. They will be mixed, pooled, and so there's the pool, and they'll be simultaneously subjected to mass spec analysis. And it isn't until they get into this collision chamber that those coloured tags come off, and that's what's counted for the abundance. So one can think of this particular process as a block, okay? And the number of samples in that block being the experimental units in the block. Now, later in 2007, Cho and his colleagues introduced an 8-plex system. And so here I'm showing you, and for the rest of this talk, I'm going to talk about blocks of size 8 at phase 2. Okay. So what are some of the things that we need to worry about when designing both the phase 1 and the phase 2 experiments? What has been found in the human liver is that there are transcriptional differences. There are differences at the transcriptional level in different regions of the liver. It's not entirely clear if this is the case with rat livers, but certainly given their shape, the lobes, it's anticipated that they might be different, and our initial pilot study suggests this is the case. But these differences in transcriptional level between lobe types is not of primary interest. It really is of secondary interest. The other thing we want is to at least achieve orthogonal factorial structure, so in other words, have no aliasing between our main effects and interactions that involve diet, surgery, and time. So what we can do is we can construct a row column design at phase 1, where our animals are our row blocks, the biopsy order are our column blocks, and these are of course going to be confounded with time. Now what we can do is we can arrange the lobes in a Latin square, and if NL represents the number of lobes in our study, and R is the number of, R is the replication, then we're going to have R times NL animals per treatment arm, okay? Now R here is an integer. R is an integer, a positive integer. So it says we're going to have, if I want to achieve this orthogonal factorial structure, then I need to have integer multiples of the number of lobes and equivalently the number of times. So if I want one complete replicate, then R is 1. So what we have here is the analysis of variance table looking at the decomposition of the information about our main effects and our interactions. Now I know these are big tables, but the couple of things I want you to notice, here is our single replicate design and here's our 2 replicate design. What we notice here is that some of our interaction effects are split between 2 strata. And perhaps one of the most interesting things is that although time and lobe on their own occur in that bottom stratum, those main effects, the interaction is split 20 degrees down, 20 degrees of freedom down in this bottom stratum, and 5 of them move up to the top stratum. And when we look at the design, it should be clear why this is the case. What's nice here is that this 4-way interaction could be used as an estimate of our error, but this would only be the case if we really knew that this interaction were negligible, and we don't. If we use 2 replicates, then what we have, with a 2-replicate design, well, we have pure error, and it's great. We have 24 degrees of freedom here between animals to test our main effects of interest, but we have 120 degrees of freedom down the bottom stratum, and it's more than enough. It's more than what we need, and these experiments are enormous to conduct. Something like this, where there are 36 how many animals do I have here? I have 6 twos, 24, 48 animals. That's 48 days of surgery for the surgeon to do these liver biopsies. It's a lot of work. So, the one replicate design, it's large. It involves 144 biopsies. It has 20 residual degrees of freedom in the intra-block stratum if the four-way interaction is negligible, but we don't know that this is a reasonable assumption. The two replicate design, this is far too large to be managed and very expensive. There are too many residual degrees of freedom, but of course it affords us the benefit of being able to test for the significance of the four-way interaction. But it's just too resource expensive. So, what I want to think about now is how do we take this material and assign it to our blocks of size 8 at phase 2. But in order to do this, I want to scale down a little bit. Rather than looking at 6 times and 6 lobes, I'm going to reduce it to 4 times and 4 lobes, just to keep it a little simpler and to be able to show the design, display the design on a screen. Okay. The second phase experiment, the things we need to consider is that we are harvesting the tissue the harvested tissue has to be processed in the laboratory. This requires a lot of additional steps in handling of the material, pipetting, extracting the protein, total protein, all that sort of stuff. So there's a lot of variation that's actually introduced at this stage. So we actually need to be concerned about these additional sources of variation that are introduced at phase 2. And if we have, as I already mentioned, if we have one replicate, we'll have 144 samples. If we have 288 samples. We want to work with something in between. And we're going to consider only 8 plex ITRAC or isobaric labels, so 8 proteomic samples, 8 proteomic samples per mass spec run. And so this is going to involve somewhere between 18 and 36 runs. So I'm still talking about the full-scale experiment here, but in the next slide, I think I'm going to the smaller one. So this full-scale experiment could cost us somewhere between 63,000 New Zealand dollars and 126,000 New Zealand dollars. And so our dilemma is how do we statistically efficiently allocate samples to runs? So in our scaled-down problem, we have 24 animals, 6 per surgery by diet group. Now, instead of having an integer multiple, R is 1 or 2, we're looking at R equals 1.5, and that's why we have 6 animals per treatment arm. Now, we considered 3 possible designs in terms of assigning the biopsies to the different orders. So here, what we've done, the 4 factorial treatment combinations, are these: sham chow, sepsis chow, sham obese, sepsis obese. There are 6 animals labelled 1 to 24. And what this is saying here is that for animals 1 to 4 and 13 to 16, basically we're going to assign those lobe types in a Latin square, a circular Latin square. So we can see A, B, C, D. Now we shift to the left one and A wraps around. And that pattern continues all the way through. An alternate approach is to use this nested Latin square. So we can see these little Latin squares of A and B, and of C and D, and then the green and the blue. And another one, which turns out to be probably not a very good idea, is to confound the lobes with A and B with order 1, 2, and C and D with order 3, 4. Now, just as a little aside, this slide here really is this is the linear model or linear mixed model for the phase 1 experiment, and it's really here just for the notation. And the thing I really want you to take home from this is that the animal random effect, I'm using the variance component, or the symbol sigma A squared for the variance component, and for the order effect sigma 0 squared. And if we could measure the thing that we want to measure, protein abundance at this stage, we would have this sigma squared down here, okay? So this is within animals and order. Now the symbols here, these really represent, it looks like an S for surgery, delta D for diet, tau for time, lambda for lobe. So that's all those symbols really are there for. And what I'm doing here is I'm just simply going to use the harmonic mean of the, so I'm going to compute the average efficiency factor, which is the harmonic mean of the eigenvalues of this matrix here. So C here is a contrast matrix, and A is the inverse of the information matrix, the generalized inverse of the information matrix. So basically, I just need some sort of criterion for assessing where is my treatment information going, or where is the information on my treatment contrasts going in my analysis of variance table. So what we see is that if we have to use a non-integer multiple of, or a non-integer value of R, we get lots of mixing up of information. So the analysis of variance table is so big I've had to split it up onto 2 pages. On the left side is the circulant, the results of the circulant design. On the right, the disconnected design. I've not looked at the nested Latin square because it's very similar it behaves in a very similar way, but not exactly the same to the circulant. And so what we see is that we get this really nice, property, and I seem to have left my variance off here, but we get this nice property where we get the variance, the part of the expected mean square that's related to the random effects of our variance components is the same all the way through. And we should have a VA down here, apologies, and down here, where VA is sigma squared plus 4 sigma A squared. So all of that part works out very nicely, but what's not very nice is that we're actually getting some of the other effects leaking in. So we're getting some contamination of our estimates by sometimes higher order interactions, sometimes much worse. Notice that we have 8 degrees of freedom here for our residual from the circulant, and 16 here. However, we're getting a funny split of our degrees of freedom for some of the effects that we're quite interested in. In fact, what's really going on is we're getting aliasing of effects, and some of these things simply can't be estimated. This is the second part of the table, and it's not very legible, but essentially you can see that we're getting different bits appearing in these different types of designs. Perhaps what's more interesting here is to go to these tables of efficiency factors, and so I'm able to fit the whole table up here, and what I can see is that, well, what we can see is, for example, here, that surgery is estimated, the main effect of surgery is fully efficient. All 3 degrees of freedom appear in this stratum, but there's some leakage from that 4-way interaction, and it's 13% of that information. So that's basically what these tables are telling us. So one of the ideas I'm trying to get across here is that we need to think about what's happening at phase 1 with our phase 1 experiment, and when we take that information and put it into a phase 2 experiment. And I need to really rush. So basically, what we did was we tried 2 different approaches. We took our animals from our phase 1 experiment and we confounded them with the runs at phase 2. So you can see here we've got animals 1 and 2, 3 and 4, 5 and 6. So we've got confounding of animals both with tags and with runs. An alternative approach is to use this Latin square arrangement, and again this is one of those nested Latin squares. And so what we wanted to do was look at what happens to the phase 2 analysis. Now, I don't expect you to look at this. All it's here to say is we have a package called InfoDecompute, an R package that'll do this decomposition for you. And I'll quickly move that away. It does the decomposition for you, and in fact it'll provide the LaTeX code that generates these tables if you so want. So what are some of the things that we see? Well, at phase 1, what we saw was that for the circulant design, we had 8 residual degrees of freedom between animals, 24 within animals dot order, and of course many more for the disconnected design. What happens is when we have to consider runs and tags from our second phase proteomics experiment is that we lose a lot of our residual degrees of freedom if we use this confounded type of allocation. But we managed to actually recover lots of our degrees of freedom or retain as close as possible to our original degrees of freedom if we use this nested Latin square design with our circulant design. In addition, we see that our variances remain the same if we use this nested Latin square, so we're getting basically the same, estimating the variance with almost the same efficiency, whereas here we've got confounding with runs and we've got this additional noise due to the run variance component. So we really do need to worry about how we allocate our samples from phase 1 to our phase 2 experiment. And these here are simply showing, well, what happens to our information. And we can see surgery by time, for example, is estimated fully efficiently in these strata, but they're tagging along lots of bits of other effects. Ultimately, we're better off in this space here, essentially, but we've got so few degrees of freedom. Okay. So ultimately, I keep saying this, what we need to do is we need to be concerned about or give consideration to our phase 1 experiment, the properties of our phase 1 experiment when we design our phase 2 experiment. What our phase 1 design does do for us is it provides us with an upper bound, or at least a raw upper bound, for the residual degrees of freedom available to estimate our error. I say raw because if, for example, we were to use REML, we may be able to recover some more information and increase our effective degrees of freedom. The trouble, of course, is that currently we're manually designing these experiments and we have some ideas about what works and what doesn't. But what we want to do is explore different objective functions. What we found is that we need to actually be most concerned about our treating our experimental unit as a treatment in assigning it to the phase 2 blocks. And the reason for this is, especially in this context, it turns out that the experimental units are carrying with them their treatments anyway. So for the most part, what happens to the treatments will be defined once we allocate our experimental units to phase 2 blocks. The process is actually quite slow when we're trying to apply simulated annealing, so we need to consider what might speed this up, because although they look like small experiments, they're actually quite large. And that's about all I wanted to say today.
We have time for about one question at the moment, but I'm going to make a rule. You can only ask it if you're in the front half of the theatre so we can hear it. Okay, well, would you join with me in thanking Cathy again, please? Thank you. Our next speaker is Dave Woods from the University of Southampton.
Okay, well, thank you very much, Ken. Thank you for the invitation. Thank you everyone for coming along. So I'm going to be talking today about some work we've been doing on how to essentially facilitate the computation behind Bayesian inference for computationally expensive models using some ideas from design of experiments and particularly computer experiments. So I'm going to start by describing the basic area we're looking at, give some motivation for these types of models, and in particular, think about the case study which we're going to be talking about, which is estimating the physical properties of human embryonic stem cells. The methodology is going to be looking at how we can calibrate these computationally expensive models, or as we'll call them, simulators. So that's how can we estimate the unknown parameters in the models, using Bayesian methods and Markov chain Monte Carlo, where we use some ideas of adaptive design and building statistical emulators or surrogates to replace, to a greater or lesser degree, the simulator in the computation. And then we'll go back to our stem cell case study and see how the methodology works. So this is joint work with Anthony Overstall from the University of St Andrews, I nearly said Southampton, he used to be at Southampton, and it was published last year in Biometrics. So in all areas of science, not just the engineering sciences now, but the biological sciences and the social sciences as well, scientists are using complex mathematical models which are encapsulated in computer codes to try and understand the physical systems which they're interested in. So we're going to call these mathematical models, and particularly the implementation of them, we're going to call those simulators. And perhaps a canonical example from engineering would be a model which is the solution of a set of PDEs which is solved using finite element methods, or more recently and perhaps more trendily, sort of agent-based simulators which build up the behavior of the system from that of the individual parts. However we set our simulator up, we're going to think of it from a statistical point of view as essentially a black box which maps the inputs and to the outputs y. So it provides for us a prediction of the outputs of the system for any value of the inputs we want to give the model. And as well as the inputs, the model also depends on these parameters, psi. So these are going to be the parameters that we need to estimate in our model. So when we want to try and use these simulators with physical data, we typically embed the simulator in a statistical model. So we actually have our real observed response y, for example, might be equal to the computer model, the simulator, plus some error term. In this talk, the error term is going to simply account for the random variation we expect to see with physical data. In other situations, that error term might also try and account for some systematic error between the computer model and reality. So our basic problem is we want to learn about these parameters psi from our noisy data. And why is it? The reason this is difficult is because however we decide to do our inference using maximum likelihood or Bayesian methods, we're going to have to make very many evaluations of the simulator, and often these are computationally expensive, so it takes time to do this. So if we're doing Bayesian inference, this is going to be through evaluations of the unnormalized log posterior, for example. So the standard solution to this problem now is to use a computer experiment to build a cheap approximation or surrogate to the simulator as a function of these unknown parameters phi. So a computer experiment, we essentially choose or carefully choose a subset of the parameter values at which to evaluate our simulator, and then we use the resulting simulator outputs with those inputs to build a statistical model to act as an emulator or a surrogate to the simulator. The most typical form of emulator is based on a Gaussian process model, which has a nice non-parametric form and is able to adapt to the form of the simulator. And then, as I mentioned before, to some degree or another, we replace the simulator by the emulator in our inference and calibration problem. So the example we're going to look at is involves stem cells and how we can estimate the physical properties of stem cells. So the underlying scientific hypothesis is that stem cells can be classified into different groups according to their physical properties. So, for example, it could be that these groups correspond to the therapeutic advantages that different types of stem cell have. Turns out that the physical properties that we're interested in, which are listed here, so the refractive indices of the cytoplasm and the nucleus, and the radius of the cell and the nucleus, it turns out these properties aren't necessarily that easy to measure directly. But they can be estimated from what's called an optical trapping experiment. And these are the types of experiments that we were working on with our collaborators from the Optoelectronics Research Center at Southampton. So the way a laser tweezer or optical trapping experiment works is that if we have our sort of toy cell here, a laser beam is fired into the cell at some distance from the center of the cell, and this beam actually exerts a force upon the cell and essentially drags the cell towards the beam. So in the experiment, we can control the focus position of the laser, and then we can measure the drag force that's exerted upon the cell. And for each cell that we have, we can repeat this experiment several times. So if we do that, this is the type of data that we collect. So this is data from 8 different cells, and for each cell, the experiment was repeated 5 times. So what we've got here on the x-axis, we have the focus position of the laser, and on the y-axis we have the drag force, which was observed. So when the laser is at the center of the cell, there's no drag force observed. As we increase the drag force, or at least increase it in absolute value, initially there is an increase in drag force, or absolute drag force, until eventually, as the focus position moves far enough away from the center of the cell, the drag force starts to drop off again. And so we can see that all 8 of our cells display essentially the same behavior, but with some differences in shape, and particularly some of this tail behavior is different for different cells. And the hypothesis is that this, any changes we see between cells is due to the physical properties. So our collaborators in the Optoelectronics Research Center built a mathematical model, a simulator, which links the focus position of the laser and the physical properties of the cell to the drag force. So in our previous notation, our model now, our simulator, is a function of these unknown parameters, which are the physical properties of the cell, and x, which is the focus position of the laser. So if we simply wanted to predict drag force for a cell which we knew the physical properties of, then this would be easy. We could just use the simulator to do this, and that would essentially is the forward problem. But we, of course, don't know these parameters, and we want to estimate them from our data. So this is an inverse problem. And evaluation of the simulator in this case is reasonably computationally expensive. It takes about a minute to evaluate it for any particular set of data. So it's not ridiculous, but if we want to do this very many times, it does take a considerable amount of time. So if we wanted to do a large MCMC sample, it will take us days to complete this. And we want to be able to do this routinely for lots of different cells. So that kind of sets everything up and describes our problem. So if we then try and be a bit more general, what we're going to have is we're going to have n observations held in the vector y from some distribution which depends on some parameters theta, and our parameters theta are going to be split into q parameters of interest, which we'll hold in psi, and the rest of our parameters will be nuisance parameters, and these will typically be parameters in our variance structure. So we'll assume that this distribution depends on our parameters of interest through our simulator. So if we want to evaluate the distribution, we have to evaluate the simulator. In the problems we'll think about today, that'll be through the mean function of the mean function of our response being proportional to our simulator. And again, x is just holding the explanatory variables which we can control in the physical experiment. And of course, if we're being Bayesian, then inference for all our parameters depends on our posterior density, and we'll just label the unnormalized posterior as capital U. So, as we've said, if we want to evaluate the posterior, we have to evaluate our computationally expensive simulator, and so to avoid this, we want to construct a statistical approximation or emulator just using a few hundred simulator runs which are carefully chosen rather than the many thousand simulator runs we'd need to do for Bayesian inference. And then once we've built our emulator, we can replace to some extent the valuations of the log unnormalized posterior with our emulator. So now we have essentially a choice we have to make on about what level we do our emulation, our approximation at. So previous literature on building emulators adaptively, sequentially, which is what we're going to do here, has focused on directly emulating the log unnormalized posterior. So we have an output dimension of 1. We're just emulating a scalar. We're trying to approximate a scalar. But we have an input dimension of p, the total number of parameters. The sort of canonical approach to emulation, which comes out of the seminal paper of Kennedy and O'Hagan, is to emulate the simulator as a function of both our unknown parameters, psi, and our physical covariates, x. So again, we have output dimension 1, but now we have input dimension q, the number of parameters in psi, and k, the number of covariates. The approach we're going to take, which was also adopted by Higdon et al. In a static inference, in a static construction of emulators, is to emulate the n-dimensional vector of simulator outputs that we get by varying phi for all values of our covariates. So if we have an n-run experiment, this is going to be an n-dimensional vector, and it has input dimension q, which is clearly less than q plus k, and is also less than p. If we emulate the simulator rather than the log posterior directly, this allows us to actually make predictions from our calibrated model, and that's obviously useful in itself, but particularly it's useful because it allows us to check the statistical model that we've formed around our simulator. And by emulating the multi-dimensional output, multivariate output, we actually reduce the input dimension. And if you know anything about Gaussian process models, you'll know they scale very badly with the number of inputs, or at least they scale very badly with the number of experiments or the number of runs you have to perform, and we can perhaps do fewer runs if we have fewer inputs. So that's our thinking there. So this is the basic algorithm that Rasmussen suggested back in 2003 for adaptive construction of emulators for MCMC problems. So the first step is to evaluate our simulator for our initial design in the input space, so initial collection of values of either phi or phi and x. Then we construct our emulator using this initial set of points. Then we use this emulator to sample from our posterior using MCMC to augment our design. And then we use the new simulator evaluations from these augmented points to build a new emulator. And we keep repeating around this loop until either we decide our emulator is sufficiently accurate, or perhaps more often, if our computational resource has been exhausted. So the really important point here is that at step 3, when we've got an existing emulator and some new points, some new evaluations of the simulator, we can check the adequacy of our emulator there by using these newly sampled points. And that's really important to make sure our emulator is actually sufficiently accurate to work on our inference problem. And then when we get to step 5, we can use our emulator in our subsequent MCMC inference. So I'm going to focus, just say a little bit more about steps 2 and steps 3, which are where we've made our methodological contribution, really. Well, step 2 is quite straightforward. We just have to decide what emulator we're going to use for our multivariate response that we've got here. So we're going to do essentially a 2-stage or a hierarchical emulator where we first of all use principal components to construct a set of basis functions. We'll call these B for our multidimensional response. And then we have our principal component weights, which we'll call W, and these we model as a function of phi, using Gaussian processes. So the only real decision we have to make here is how many principal components, how many basis functions we should have. We typically don't need very many principal components in order to describe most of the variation in our response. So in our stem cell example, using between 5 and 7 principal component weights explained the vast majority of the variability, and means that we have to actually end up estimating somewhere between 5 and 7 Gaussian process models. The third step is how we actually sample subsequent points to add to our design. So Rasmussen used hybrid or Hamiltonian Monte Carlo, where the proposals are the solution to a set of dynamic equations which are discretized, and we have a step length L and a step size delta. HMC is very good for constructing adaptive proposals which give you low autocorrelation and high effective sample size. So from a design of experiments point of view, this means we have good space filling properties in our parameter space. But they do require a lot of evaluations of the likelihood or the unnormalized posterior, and particularly particularly they require a lot of evaluation of the derivative of the unnormalized posterior. In a more recent paper, Fielding extended this methodology to include parallel tempering to try and avoid getting stuck in local optima, so we can explore multimodal distributions by using lots of MCMC chains where we temper the chains, so when T gets large, the density gets very flat and it's easier to move from one mode to another. So the problem with both these methods is they do require the selection of quite a lot of tuning parameters, and if our simulator is expensive, it's not necessarily going to be the best use of computational resource to spend simulator runs tuning these systems. So we, in our approach, replaced Hamiltonian Monte Carlo with Gibbs sampling with the adaptive projection metropolis sampling scheme to sample from the conditionals which weren't available in closed form. This effectively has no tuning parameters, although we obviously know that Gibbs sampling tends to have higher autocorrelation, and so to reduce the autocorrelation, we actually thinned our samples. So we do introduce one tuning parameter, which is the degree of the thinning that we do. For the parallel tempering, we employed the adaptive parallel tempering scheme that was recently been suggested, which chooses temperatures to achieve a target acceptance rate by using the discarded iterations from the MCMC. So basically what we're saying here is there are two kind of steps. One is we are definitely going to emulate the simulator so we can do model checking, and then we want to try and reduce our dependence on tuning parameters by adopting these alternative methods. So let's just see how these work on one or two synthetic examples, and these are actually two examples which Fielding et al. Used in their paper. The first is a two-dimensional distribution with sort of banana-shaped contours, and the second is a high-dimensional example which has two distinct modes. Now, there is no simulator in these artificial examples. There's only the density, so we will here approximate the log posterior directly, although when we come back to our stem cell example, we will emulate the simulator. And we can compare the true densities and the approximations we get using the various methods using the Hellinger distance. So for each example, we have an initial design which has 50 points, and for our parallel tempering, we're going to use 5 temperatures, and then we'll do 30 iterations of the algorithm to add 150 different points. So these 50 points, initial points, are chosen using a maximin space-filling design in the parameter space, and then the additional points are going to be chosen either using HMC and parallel tempering, or Gibbs sampling and adaptive parallel tempering. And we've done 20 implementations of each of these different approaches with different values of the tuning parameters. So for HMC, that's different values of the step size, and for the Gibbs sampling, it's different degrees of thinning. And then we repeated each of these implementations 100 times. So this is our first example, the banana-shaped PDF that we're trying to sample from. And these plots show us our results. So here, what we have, this axis gives us the degree of thinning, number of iterations that we discard, or number of iterations between those we accept. And this gives us the value of log delta for the HMC. And then here we have the median Hellinger distance across our 100 iterations. So the blue are the results from our method using Gibbs sampling. We can see actually they're very, very robust to the degree of thinning. The red is the results from the HMC method. So this works extremely well. Very low Hellinger distance is at least as good as our methodology when you tune it correctly. If you don't tune it correctly, then actually it can perform quite poorly. And this dot here, this filled-in dot, which you might be able to see, that's the default from the R package which Fielding et al. Provided for their methods. This plot shows you the computation time that's required for both methods. So obviously ours increases as the amount of thinning increases, but in fact, as we can see, we don't really need to do very much thinning in this example. And the HMC method is pretty static in terms of the amount of computation. But of course, there's no simulator in this problem. So this computation is purely in terms of the sampling that we're doing in the MCMC. And if we had a simulator involved that was expensive, that computation would outweigh any computation that's going on here anyway. This is the second example. So we've just plotted here a projection into the 2 dimensions which show the 2 different modes. It's 7-dimensional in total. And then we have the same plots which give you basically the same results. Our method, the Hellinger distance is fairly constant to the degree of thinning, whereas, again, the tuning parameter in the HMC is really quite critical to its performance, and the computation performs similarly to the previous example. So I think we have covered all of those. So let's go back to our stem cell example. So here's a reminder of our data. We have our laser focus position, and we have our drag force. So we're going to fit a model to each cell to try and estimate the physical properties of each cell. So Yij will be the jth response from the ith experiment on a particular cell, and our initial model just assumes that Yij is modeled by the simulator plus a random error term. With our error term, we assume that observations from the same replicate may be correlated, and the correlation between them depends on the distance between the focuses. And our prior distribution was a truncated multivariate normal prior, which takes account of the dependencies between the different parameters and was elicited from the scientists we were working with. So now we will emulate the simulator to get the computational advantages and to allow us to do model checking. We'll do parallel tempering, so we'll have 5 temperatures. Our initial design is again 50 points, this time sampled from our prior, so we get the constraints in there. And we add out again 150 evaluations to obtain our simulator. Emulator rather. Once we have this emulator, we generated an MCMC sample of size 25,000 from the posterior of both our parameters of interest and our nuisance variance parameters. And using this sample, we can also sample from the posterior predictive distribution. So what we can see here, this is for one particular cell. The black dots are the actual data, and the gray dots are a sample from the posterior predictive distribution. So our predictive distribution is not terribly precise, and it actually doesn't seem to capture this tail behavior we see in some of the replicates terribly well. So this behavior is actually due to an error in the resetting of the lasers, but we'd like to try and incorporate this into our model. So this is our second statistical model, where we extend our model by having this shrinkage term here, which lies between 0 and 1. We give this a prior distribution, which is has a mean, which is inversely proportional to the magnitude of the focus position. So this means that when our focus position is small, around here, then we don't have much shrinkage, and we can have more shrinkage as our focus position increases. So using this model, we add in 100 extra evaluations of the simulator to build a new emulator, and we use this new approximation to generate a new MCMC sample. So this is the step you couldn't do if you were just emulating the log posterior, because you wouldn't be able to do predictions from the model. So this is the results from our new MCMC sample. We've got a much more precise posterior using this model, and it explains this tail behavior much better. So we're much happier with the results that we're getting here. Don't know how well you can see these plots. Obviously, the predictions are interesting, and they allow us to check the model, but we really want to find out something about the physical properties of the stem cells. The very flat lines here are our priors, which were really quite diffuse. The dotted lines were the posteriors from model 1, which we decided was inadequate, and the much sharper posteriors, and indeed shifted in some cases, are the posterior densities for model 2. So we've learned quite a lot, at least about this particular cell, from our data set. And we can look at these things bivariately as well, because remember, one of the things we're interested in essentially is, can we tell these cells apart from each other? So here we have samples from the posterior distribution of the parameters plotted in 2 dimensions. So this is the 2 refractive indices for the cytoplasm and the nucleus, and these are our 8 different cells. So this is cell 8, for example, and I think this is cell 4. And this is the same plot, but for the radii rather than the refractive indices. And what we can see is that definitely some cells do look different in physical properties from the other cells, and there seems to be greater intercell variability than there is intracell variability. Okay, so to wrap up then, we've looked at how we can do MCMC inference for computationally expensive computer models using emulators based on Gaussian processes, essentially. So we've used Gibbs sampling with adaptive rejection metropolis sampling to reduce the amount of tuning that's required to try and improve the computational efficiency. And we've emulated the simulator rather than the unnormalized log posterior, which allows fast prediction and, importantly, the model checking. So in our stem cell example, we were able to quickly estimate the physical properties of all the cells that the scientists had data for. We could check the adequacy of the statistical model that we were assuming and improve it, and it gives us the potential now perhaps to go on and classify stem cells using their physical properties. And there we've implemented these methods in an R package called BayXComp, which is available from either myself or Anthony if anyone's interested. Thank you very much.
Any questions for Dave?
Hello, Robert Feff from INRA France. I just a small question concerning your first 50 points. How do you choose and what kind of experimental design? And is it sensitive to this design?
So we've just been using either space-filling designs for those first 50 points. So in our artificial example, that was a maxi-min space-filling design. Or we've just been sampling. In our stem cell example, we wanted to take account, there were some quite intricate constraints between the parameters, so to take account of those, we just sampled from the prior rather than try to build the constraints into space filling. But you could try and do something cleverer with a constrained space filling design there.
No one else? Okay, well, please join with me in thanking Dave once again. The third speaker in the session is Alison Smith from the University of Wollongong in Australia.
Okay, I'd like to start by thanking Ken very much for proposing this session and for giving me the opportunity of presenting this work. It's joint work, as you can see, with Dave Butler, with Robin Thompson and Brian Cullis, and it's work that we're very much involved with right at this point in time. There are experiments happening as we speak back in Australia. Well, maybe not, they're sleeping at the moment, but fairly shortly they'll be happening. Another big thank you to the Grains Research and Development Corporation of Australia for their ongoing financial support of biometrics within Australia. And as I say, this is work that's happening right at this very moment, and it's connected to a project, and I'm going to talk about a small portion of that project today. That project relates to a series of variety trials that are happening across Australia, and you can see the locations here. They span the entire growing region of the country, which is pretty substantial. And the aim of this project is, unlike a lot of the work that I've been involved with, which is dealing with the trait of grain yield, this is now looking at quality traits. So traits concerned with the flour that is made out of the varieties, and also then the dough and then baking characteristics of end products. And It's a unique project in that we're really trying to get a hold of accurate genetic estimates. We haven't had the data to be able to do this before, and I'll explain that a bit later. And also look at things like variety by trial interaction. As you can see, because it spans the entire country, there's quite potential for varieties to perform quite differently across the country. As Kathy was talking about earlier, the traits of things like flour yield and dough and baking characteristics, they're all what are called multi-phase traits. And in our project, that's going to involve either 2 or 3 phases. The first phase for any of those traits is a designed field trial in which a number of varieties are grown in a replicated trial. And ultimately, those plots are harvested to give us the grain that goes on to be used, processed in the laboratory to obtain these traits. Just a quick look in this project, they're all relatively small trials because quality testing is very expensive, and once again, we'll talk about that later. The number of varieties only ranges from about 18 to 22. There are 3 replicates for each of the trials, and they're laid out in rectangular arrays of rows and columns. So a typical trial is this one, which is from Western Australia. We have 9 rows and 6 columns. The replicate blocks are aligned with pairs of columns. And you can see here, this is the randomisation for the varieties, and in fact the actual harvested yields for the individual plots are shown up there. And they range from about 5.5 kilograms per plot up to over 8 kilograms per plot. So a fairly standard looking experimental design. And a key thing with these multi-phase designs at present anyway is that the design for the first phase, the trial, is done separately from the subsequent phases. Okay, the second phase for obtaining the trait data involves some sort of process in the laboratory where the grain samples that we've obtained from the first phase field trial are taken. One of the examples that we're going to look at today is the thing called flour extraction or flour yield, which is, as the name might suggest, it's the amount of flour that you produce from a given quantity of grain. The process that's required for flour extraction is obviously once we've got the grain from the field trial, and that's illustrated by the first 2 pictures there, the grain is put into this contraption here. It's called a mill. This is actually a laboratory mill, so it's not like a commercial mill. It's specifically designed for milling small quantities of grain. The grain gets put through the Buehler mill, it's processed, and at the end we get some flour. And from that we can measure the weight of it, which is our flour extraction. Other examples, other traits might require another phase, a third phase. Once again, it's another laboratory process, but this time the process starts with the flour, which was obtained from the second phase. And a key example are traits to do with the strength of the dough. And once again, nice representation here. We're starting with the field trial, we're getting the grain, it goes through the Buehler mill, which produces the flour, and then from that, for this particular trait, it's actually made into dough, and it's put on a machine and it's stretched, and the resistance and the strength is measured. So there are a lot of processes involved in measuring quality trait data, a lot of machinery, a lot of manual intervention, a lot of potential for non-genetic variation. And I'm reluctant to call it error variation because it gets the people in the laboratory very upset. So we'll just keep with non-genetic variation. But in the laboratory, certainly for flour extraction, the mills are things that warm up during the course of the day, so you can get slightly different results at the end versus the beginning. So with all this kind of knowledge, obviously what we would like to be able to do is help to create a design to accommodate some of that variation. And by a design, obviously I mean things like we want to be able to randomise, we need to have replication, and that's replication in all the phases. And if for people who wish to see some more details on this, there are 2 key references, or quite a few, but One includes a paper that we wrote in 2006, and a lot of the work that Chris Bryan and Rosemary Bailey have been involved with. We obviously have replication in the field. We have multiple plots of each variety. In terms of the first laboratory phase, we've taken, from the field, we've got our bags of grain, and literally from a particular bag of grain, we split it typically in half, and then that gets processed separately in the laboratory. So for our Western Australian trial, where we've got 54 field plots, if we were to do full replication in the first laboratory phase, so full replication to do the milling, that would require 108 samples. But similar to Kathy's problem, quality testing is incredibly expensive. In the project that we're doing at the moment, to do the full range of quality tests on one sample of grain, it's over $650. And that's a rough conversion rate to euros. It's incredibly time-consuming. Milling, using that Buehler mill, they can only mill 5 samples per day with one mill. We're actually going to use 2 mills in this project. So milling that number of samples for even one trial is going to take 3 weeks. So for 12 trials, it's going to be 3 months. And then there are a whole range of other tests going on, so the whole thing can blow out quite enormously. So obviously full replication in all phases is not something that we can think about. So we've come up with a strategy for our project based on the cost of the project and the budget that we had, and the laboratory throughput for all these tests, and a whole lot of other complicating issues, including the fact that this laboratory has never been involved with experimental design work before, so they really had no idea about replication or indeed following a given order of processing. So there have been many challenges along the way for this one. But the limits that we've set up, we're going to take 33 bags of grain from a field trial. We're going to split 7 of those to make 40 samples of grain for milling, and then we're going to, once that's milled to produce flour, we're going to split 5 of those to go on to do dough strength testing. And the way that we might go about reducing replication in the laboratory, some work that we did previously was something called a PQ replicate design for 2 phases. And all that was happening there was that for our set of varieties, for our 18 varieties say, then a proportion of them we're going to test using 2 replicate field plots, and the rest of them we're just going to use a single field plot from the field. Having chosen those plots that we're going to use, then a proportion of those gets split for replication in the laboratory. And we've shown in papers and with a lot of work that we've done that gives us enough replication to be able to apply appropriate linear mixed model analyses of the data. So we could use that approach with our data. It will allow us to estimate the effects we're interested in. But and so for our example with the 33 bags of grain from that particular trial where we had 18 varieties, we'd have 2 reps for 15 varieties and then just a single plot for each of 3. So we would have replication, but clearly we're wasting an awful lot of information. We had 54 field plots and we're only going to use grain from 33 of them. And in some of the larger trials, that loss is obviously greater. So can we do better? Well, obviously we think we can. And the way that we're proposing to do that is to, once again, have some of the varieties, we're going to do individual field replicates for some of the varieties, and for the other varieties, we're going to use composite samples. So literally, we take grain from a number of plots, they get mixed together in a bag to produce a composite sample. So in that way, we can use information from all of the field plots rather than just a sample of them, or a subset of them. And once again, we can apply linear mixed model analyses for this, although it is a bit more complicated because we're going to have non-standard design matrices arising from the fact that we're compositing samples. In the paper that we've just written, in fact, just been, or is about to go into press, we've shown that by using this mixture of composite and individual plot samples, we get more accurate predictions of our effects of interest than we would if we use the PQ idea, particularly if the between plot variation is large compared to the lab variation. And the graph that I've got up here shows on the x-axis we've got the plot variance ratio, so plot variance relative to lab variance, and on the y-axis is effective replication for 4 different compositing strategies compared to a PQ replicate design. So when there's hardly any plot variation relative to the lab, then there's not much gain in using the compositing approach. But when there's quite a substantial amount of field variation, the gains of using compositing strategies can be quite substantial. We've got up to 40% gain in effective replication on the graph we've got there. So just what am I talking about with these strategies? Well, the strategies that we had up there involved 3 different types of compositing. I've got what I've called a C3 composite sample, which is a composite of all 3 field plots. A C2 is a composite of 2 plots, and a C0 is, a composite of 1. It's an individual plot. We're not mixing it with any other plots. So if we look at 3 of the varieties from that trial that I showed you, the variety frame is going to be tested as a C3 sample. And the way that you might be able to think about this is that the composite is derived from 3 potential parental plots. For the variety frame, it was grown in 3 plots. If I went back to the field diagram, I could show you that it was grown in column 1, row 2. So obviously the C notation is for the column and R for the row. It was also grown in column 3, row 1, column 6, row 5. And the individual weights for those 3 plots is given up here. And the composite, literally, we get those 3 bags and put them into one larger bag and mix it very thoroughly. And we end up with a bag that weighs nearly 21 kilos. The variety Jance is going to be tested as one individual replicate sample. So the plot in column 5, row 7, which weighed 6 and 6.1 kilos, it's going to be tested as its own, as a single entity, whereas the other 2 plots for that variety, in column 2, row 6, column 4, row 2, they're going to get composited together. So it has 2 samples going through for testing. One's a composite of 2 plots and one is an individual plot sample. The variety Yitpi is going to be tested with 3 samples, each of those corresponding to the individual field plot from the trial. So to get your head around designing for multiple phases, we've come up with this notation. It's not traditional sort of notation, but it captures the idea of experimental units within phases rather than just overall, because it's a complex business moving from one phase to the next. So I'm going to talk about treatment units, treatments being the actual treatments that are applied in the phase. Replication units are the units that are going to be replicated in that phase. The input units are the actual entities that are to be put through whatever process is associated with that phase and that particular trait. Experimental units are the physical units that define the structure of the process, and we're obviously going to allocate our input units to the experimental units using some kind of design. The observational units are the things at the end which are measured to obtain the trait. They might be physically the same as the input units, but typically with this sort of data they're not, because the process itself changes that unit. And the phases are linked via transitions from the observational units from one phase through to the replication units for the next. Now, just to get your head around what that jargon means, it's probably easier to look at this in the context of our example. So phase 1, which is our field trial, we had 18 treatment units, 18 varieties. They are also the things that are going to be replicated. So they're the replication units. Input units, well, we had 3 packets of seed, basically, of 18 varieties. So we've got 54 packets of seed. The experimental units are the physical plots, which are indexed by the rows and columns. We've also got blocks as a factor of interest. And the observational units are the 54 bags of grain that were harvested from that trial. The green arrow shows that the transition from phase 1 to phase 2, we take the 54 bags of grain, we do our compositing scheme to end up with 33 bags of grain. And in that second phase then we replicate 7 of those samples. We split 7 of those bags of grain into 2, and the rest are done as single samples. We're going to put them through a milling process. So the physical units there are actually the orders of processing. Milling is done sequentially. And we've also got 2 mills in operation. So things like the day, it's going to take 4 days to do this, and the order within a day are important. And at the end of that process, we get 40 bags of flour. They are then taken, follow the red arrow, they go through into phase 3 for the phase 3 testing. The reason I've put a red arrow in is because whereas from phase 1 to phase 2 we've done some compositing of the grain, in phase 2 to phase 3 we haven't done any compositing of the flour from one phase to the next, but we could do that, but life was complicated enough for these people. So, okay, so for any of the phases we want to have a design. We want to allocate our input units to experimental units. We use model-based design techniques, and the aim being to minimise the average pairwise variance of our variety effects. And numerous people have looked as that as a criteria which is suitable for the sort of designs we're talking about. And I'm going to speed up a little bit here. So we generate our designs using a package called OD, Optimal Design. And the syntax of OD is very similar to ASMLR, which is what we use for linear mixed model analysis. So to create a design, basically we have to have a starting design which sets up the factors of interest, the blocking factors, for example, and we need to specify the mixed model that we want to use in our optimisation. The model that we use is obviously similar to the analysis that we propose to use for the data at the end. So we need to put in all the sources of variation, we have correlation potentially. This is a tricky business, but a lot of Chris Bryan's work, Rosemary Bailey, has been devoted to helping people formulate appropriate models for their designs. So for our particular one, if we're looking at flour extraction, the mixed model will have random effects for varieties, for blocks in the field, for potentially rows, columns, the field plots themselves, which is important to maintain the strata going through to the next phase. In the milling phase, we've got the mills themselves, which are likely to be a big source of variation, the day on which the sample's milled, and the order within the day. If we weren't doing the compositing side of things, then in OD or ASMLR, the syntax would be, as well, I have it here, very simple. This is for the random effects. We just list the factors that are associated. If we've got compositing going on, then we end up with some non-standard design matrices, and if we want to see how we deal with this, if we look at the plot effects, if you remember the variety frame, which was a composite of 3 parental plots, the effects for the individual parents, well, I've used U as representing the effects, so it's the effect for column 1, row 2, column 3, row 1, column 6, row 5. They were the 3 individual plots. When we composite, you can think of that as an averaging process. So essentially, the plot effects for the composite sample is just the average of those 3 effects. Similarly for Jantz, when it was a composite of 2, it's the average of those 2 effects. The fact that we've got mixtures of averages of 3 things and averages of 2 things, then the way to handle that is basically to use the lowest common denominator. So we think of it as averages of 6 things. So for frame, the plot effect for that composite sample is a 1/6 of, well, we've got a double up of column 1, row 2, then column 3, row 1, and so on. Clearly, we're ending up with non-standard design matrices for our plot effects. In OD and ASMLR, we can use an AND function to do this. And essentially what we need to do is in the data frame, we need to have extra factors. And because I've got I'm averaging 6 things here, I need 6 factors going in there. So it's fairly intuitive once you see that you're trying to average 6 effects. They are the 6 factors we need to put in. And the notation is, as I've got it at the bottom here, and, which essentially is summing the design matrices for those 6 plot factors and dividing by 6. It doesn't have the notation for /6. We've got to multiply by 0.1667. So it's very clever syntax to be able to get linear combinations of design matrices. We also need a starting design, and all I want to say here is we obviously need to pull in our milling day and milling order. And ultimately, we want to balance varieties across a lot of the second phase blocking structure, mills being important. And you might have to, well, you might think you have to do that up front, but you don't. We can actually let OD work that out for us. Now, I know this looks gruesome, but it's just because I also have blocks. So the B stands for block, C for column, R for row, and P is our plot factors. So this is showing us the full model that we're going to fit and the starting variance parameters. Just to point out, the mill variance of 10, which is enormous, basically it's constraining that to be like almost like a fixed effect, so that varieties are going to be balanced across mills. And in fact, that's exactly what happens, is that for those varieties that have 2 samples, well, you get 1 for each mill, and with 3, it's 2 and 1. Okay, finally, we get to search for the design, and this is a new syntax within OD, and it's incredibly clever because we can actually optimise for varieties, whereas in previous things we would have optimised for the things that we were replicating in the second phase. We can actually get at the varieties themselves, but a key thing is we need to carry through, as we permute the varieties, there's a whole lot of plot information that has to be permuted as well, and this pipe operator allows you to do that. Okay, it's not running not too badly. All right, obviously, with the time available in the talk, there's been a lot of detail that I haven't been able to include, a lot of it, the maths behind it, but also a lot of the operational issues with dealing with this. But if anyone's interested, this is the paper that is about to appear. There's a lot of interesting issues left in this. In particular one, which is that at the moment we're optimising for varieties based on known variance parameters, and obviously in the real world, in the end we're actually going to estimate these things so that can have an impact on the design also. So finally, this method that we've put up here, it's actually going to allow us to quantify variety by interaction in these really important quality traits. It's of huge importance to the industry, both in terms of farmers knowing which varieties to grow and in terms of breeding better varieties. There have been a lot of projects over the time looking at quality data in Australia, but unfortunately they're all flawed in the sense that none of them have appropriate data. Typically, they're all based on fully composite samples, so there is no field replication, no lab replication, no lab designs, so that certainly variety by environment interaction is confounded with field, with lab, and we just can't get a grip on what's going on with variety by environment interaction. So it's really exciting to be finally able to get a data set where we can look at this. And of course, along the way, we've had to work within a lot of tight constraints, clearly the budgetary constraints, but also, as I mentioned before, the fact that the lab hasn't ever been involved in experimental design work, and so we've had to come up with a scheme and explain that scheme in terms that they can actually carry this thing out. And I'll let you know how that goes. So thanks very much. Are we sharing, Ken?
Questions for Alison?
Alison, is there any reason why you presented this as plots for varieties and lab testing for food, but is there any reason why it doesn't carry over more widely, for example, to the entire field of medical research, in which the plots are equivalent to individual people, and we're measuring in the laboratory some properties of theirs in response to some treatments?
Okay, thanks, David. Yeah, look, it has many applications where anywhere where the second phase is an expensive thing to test, then there's potential to composite things from the first phase through. So certainly medical, environmental, where they're measuring very expensive, traits like mercury, things like that. It certainly has much wider application than the field experimentation side.
That's right. The composites required because of the cost of the lab phase, but even without large cost to the lab phase, this is still of benefit to medical research, because you reduce the variance of the treatment effects by actually by averaging better over the lab allocations.
Yes, certainly. I mean, as you know, agriculture is our application, and the cost was the key driver here, but it sounds like your application it would work really well for also.
No other questions? Okay, well, please join with me in thanking Alison again. Well, I get to make a few concluding remarks. I guess the first one is that I would like to thank the program committee for making this invited session number 1. I consider it very appropriate that experimental design should be number 1, because all of those who know and believe in experimental design are very well aware that unless you get the design right in the beginning, then no amount of fancy computer fiddling and data analysis is going to give you the right answers or the complete answers. So I think it's just an additional way to reinforce that experimental design should come first. This is the first session, as I understand, ever to be recorded on the IBS website, other than perhaps a presidential address. So it's also a privilege that we get to have that opportunity. So I look forward to revisiting the session once it's made it to the IBS website, and invite you all to do so as well. I'd like to mention one speaker who had been intending to give a presentation, but unfortunately had too many logistical difficulties in getting a visa. And that's Zhengguan Zhu from Iowa State, who was going to be talking on spatial temporal design for estimating biomass and biomass change of forests. Now, that also, I think, would have been quite a interesting and different approach to experimental design. I'd like to make just a few brief remarks. I'll take the papers by Kathy and Alison together because they're somewhat more similar than different. Some of the similarities, of course, were that they were both multi-phase experiments, and it was very clear that extensive planning was needed because of the high costs of obtaining data. Now, that was not necessarily getting the wheat or what have you, but the processing and all of that was going to be very expensive. And it was clear that they were using conventional, in inverted commas, designs, randomised complete block designs, Latin squares, in part of the experiment, but only part of it, because then they had to think outside the square to get around problems. And to me, that's one of the fascinations of experimental design. And I don't think we managed to get that message out to many of our colleagues. I have a very distinguished colleague in a different field of statistics who seemed to think that once you got to three-way factorials, you'd hit all the boundaries that experimental design had ever managed to achieve. And this clearly demonstrates that's not the case. There were some differences between those papers because Kathy finished up with similar data at the end. You know, all the units were sort of the same kind of thing, whereas Alison finished up with what I would call dissimilar data because some of it was composited, if that's how it's pronounced, and some of it was not. And Kathy, I believe, would have probably had a straightforward analysis if she has a good statistical package. Now, what you call a good statistical package may depend upon which continent you come from. Alison clearly had a more challenging analysis. But I think each of them demonstrate that if it were ever true that it was the case that the experimenter had to mould the experiment to fit the design that the statistician could give them, then that's well and truly gone. Dave Wood's paper challenges, at least for me, the conventional idea of an experimental design, such as a field layout, but it certainly fits the notion of planning and collecting data to answer a research hypothesis. And I'm sure we're going to see more and more computer experiments as the years go by. While Dave wasn't necessarily trying to reduce actual monetary costs, he was certainly interested in reducing the computational cost of an experiment. And it was, to me, the most extensive use that I've seen in Bayesian methods and MC in designing an experiment, and I assume that there'll be lots more of this in the future. So I'd like to conclude and give you an extra few minutes at lunch by saying that I think this demonstrates that experimental design is not a thing of the past. The problems weren't all solved 40 or 50 years ago. I don't believe it's boring, I don't believe it's unimportant, and I'm certainly convinced that we do not have all the problems solved. So we continue to need to get innovative techniques from all sorts of fields so that we can ensure that the data get gathered, the right data with the right design so that you can answer the research hypotheses. No confounding that messes things up, and as many right answers as possible. So I'll ask you to join with me one last time in thanking our speakers, and that'll give you 10 extra minutes to get lunch. Thank you.